Hello, and a very good day to you. I'm Dr. Rayhan Falari, and it's my pleasure to welcome you back. This time around, we'll be exploring the key genetic and molecular mechanism that underpin gametogenesis. So in this lecture, we'll be understanding the formation of the male and female gametes while discussing some sex-determining genes. We'll look at meiosis-specific genes. We'll look at some hormonal regulation, some epigenetic influences, molecular regulators, and genetic defects that may impair gametogenesis. So to set the stage, gametogenesis refers to that essential biological process that is responsible for the creation of haploid gametes. And by haploid, we mean one copy of each of the chromosomes contained in that cell. And these are the building blocks of sexual reproduction. And for this process to be precise, it's usually orchestrated by a sophisticated genetic control, which is why we are having this lecture, a genetic control mechanism that helps to ensure that there is accuracy in the execution of the meiosis, the meiotic division, and in the production of the functional gametes that will eventually lead to fertilization. On this left side, you have oogenesis, where the primary oocyte transforms into the secondary oocyte, giving off the first polar body and then giving off the second polar body after fertilization. And then you have the in spermatogenesis on this right side, where you have the primary spermatocyte, which is also diploid. 2N here means each of the chromosomes there is doubled, before then transforming into the secondary spermatocyte, by which time it has become single copies of N, and then the secondary spermatocyte further divides, and then you have here they have become spermatids, right? At this point, they are known as spermatids, and then they undergo further maturation into what is referred to as sperm. So take note of these keys right here. So here is the zygote, okay, that has been formed after fertilization. So to have a further appreciation of this, we further define gametogenesis as a dynamic process that involves meiosis, which crafts the male and female gametes that are required for perpetuation of genetic information. So gametogenesis is divided into spermatogenesis in males and oogenesis in females. And the ultimate aim of the whole process is the production of haploid gametes. You see, they started as diploid, and like with body cells, like with somatic cells, that is the cell in the rest of the body, every other part of the body except for the gamete. So the somatic cells, they contain two copies, right? Every chromosome is having two copies. But for these sex chromosomes, I mean for the sex cells, so once there is once fertilization is imminent, we need for the sex cells, the gametes, to be haploid. So that's why the ultimate aim is the production of haploid gametes. So there is a time when the gametes are being diploid, so we need them to become haploid in order for fertilization to happen, such that the sperm in males and the egg in females will end up fusing in fertilization to form a zygote that will end up being diploid. So the stages to spermatogenesis is our focus in this slide. Spermatogenesis begins with mitosis, that's mitotic division on both ends, right? So you would consider here the spermatogenesis. It starts with a spermatogonia, diploid spermatogonia. And before we go into that, understand that the phases of spermatogenesis are generally three. So you have a time when there is multiplication, that's the multiplication phase. You have the growth phase, okay, and then you have the maturation phase. So the multiplication phase, at that point we have a spermatogonia, and the spermatogonia, which is diploid, undergoes mitotic division to become the primary spermatocyte. The formation of this primary spermatocyte initiates the growth phase. So this grows and then undergoes meiosis. You know, mitotic division is more like a multiplication division. That's why we refer to this as the multiplication phase. So the at this point, from the primary spermatocyte, it undergoes meiosis, which is a reducing transformation. Okay, so meiosis one, there are going to be two meiosis. So the first one is meiosis one, and this reduces the primary spermatocyte, which used to be diploid, into haploid gametes. So at this point where we have the haploid gamete, the first haploid gamete known as secondary spermatocyte, this that's the end point of the first meiosis. Okay, so at this point it's called secondary spermatocyte, and then each secondary spermatocyte, which is also haploid, undergoes meiosis 2, and that way it divides, it replicates itself, all right, to then give us what we know as the spermatid. So the end of the growth phase gives us a spermatid, which takes us into the maturation phase. So all the spermatid needs to do is to just mature as it is in that same number. The process of maturing from spermatid into spermatozoa is known as what? Spermiogenesis. So take note of the difference in spermiogenesis and spermatogenesis. Spermiogenesis is a stage in spermatogenesis. So the whole thing here is spermatogenesis, while spermiogenesis is the last stage of spermatogenesis. So the conversion, the transformation, the maturation of the spermatid into a spermatozoa is what is known as spermiogenesis. And on the other hand, you have oogenesis. Oogenesis for a female begins, of course, since fetal life. So during fetal life, the the oogenesis commences with the oogonia. We have spermatogonia, the female correspondence of which is the oogonia, which is also diploid. This oogonia also goes through its own multiplication phase through the mitotic division, and that still gives the same, I mean, a diploid gamete, which at this point is known as primary oocyte. Then the primary oocyte undergoes the first meiotic division. This meiosis 1 is completed prior to ovulation. Okay, prior to ovulation. So the primary oocyte is formed during the fetal life, right? While it's in the adult life. So these are the two main segments of the female of the oogenesis. You have the fetal life, and then you have the adult life, right? The adult reproductive life. So it's during the adult reproductive life that you then have meiosis 1 getting completed. So, and that produces a secondary oocyte. And it's on the formation of the secondary oocyte that you have the first polar body being given off. And then, so the secondary oocyte is now haploid. It's a single N, it has haploid. And then the secondary oocyte, similar to the secondary spermatocyte, now undergoes meiosis 2, right? So this meiosis 2, its completion is what gives a matured ovum, which then gets ovulated and gets fertilized and takes place in, takes part in the fertilization process. So we understand that, I mean, from this explanation, the spermatogenesis begins with mitosis, then it progresses through meiosis 1 and 2 on both sides, whether it's spermatogenesis, which is similar to what happens in oogenesis, I mean. And this culminates in the intricate process of spermiogenesis, which is the transformation of the spermatid into spermatozoa, which then transforms immature sperm into mature motile gametes. On the oogenesis, it's a parallel process that involves mitotic divisions, meiosis 1 and 2, and the completion of oogenesis, which also results in the formation of a mature egg. So the key differences: spermatogenesis and oogenesis, they share similarities, but they diverge in the timing of meiotic events. The meiotic event in the meiotic event in oogenesis, you see it happening. You see meiosis 1 is completed prior to ovulation, which is post-fetal life. So a number of the activities are completed during completed during fetal life for the female. So you have all of the eggs that a female will ever have being born with her. So at birth, you have the arrest of meiosis 1. in the female, and it only gets completed prior to prior to ovulation, so for all the eggs. Whereas for males, all of the there is none of the stages of spermatogenesis that happens in fetal life other than the migration, of the germ cell lines, or the migration of this of the testes. But ever since that has happened, the spermatogonia remains dormant in the male up until puberty, when the respective stages of the spermatogonia maturation commences. So, but in female, the bulk of the oogenetic stages happen in fetal life, and they only get completed after arrest at meiosis 1, uh, prior to ovulation. So, to further move towards the genetic, which is the genetic focus of this lecture, here is a reminder on the structure of a chromosome. So, within every cell, you have the nucleus, and within the nucleus, you have the chromosome. So, here you have two sister chromatids joined together in this single chromosome. All right. So, when you have a chromosome that is joined together at this center, at this point, it's getting ready for separation. So it's getting ready for the telophase stage of replication. So in this case, you still refer to them as sister chromatids. All right. So by the time they then separate, each will then stand as a chromosome. So each chromosome is made up of tightly coiled and wound DNA double helix, which is all made up of chromatin materials. And then this DNA double helix is wound around histone molecule. So here is histone, which is a protein that is associated with the DNA double helix. So the DNA is the hereditary material that is organized into chromatin, and which is a dynamic structure of DNA that is wrapped around histone proteins that influences gene accessibility. And the three-dimensional arrangement of chromosomes profoundly influences the expression of genes, and this helps to play a critical role in the regulation of gametogenesis. So going further to understand the genetic basis of gametogenesis, here is the cell containing the nucleus, and within the nucleus you have these chromosomes that have been amplified here, and then taking this part of this chromosome, which is made up of the DNA double helix. So you have that, you can see the ladder. So the sides of the ladder, that's the sugar phosphate backbone, and then you have the steps made up of nucleotides, right? Made up of the nucleotides, which has the adenine, the cytosine, the thymine, and the guanine. So the nucleotide is when you take the respective component of the phosphate sugar backbone with it. But these are nucleobases, each of these sites. So a segment of this DNA is what constitutes a gene. So when we talk about gene, it's referring to this. So it's a segment. So it can be short, it can be long, but it's somewhere, always somewhere along this DNA. So from here to another end is another gene. From here to another end is another gene. So these chromosomes, which you understand are composed of DNA and proteins, serve as the carriers of genetic materials and of genetic information that guide the intricate dance of gametogenesis. And for the genetic transfer to be accurate during gametogenesis, the the genes have to come into play to ensure that there is a faithful transmission of traits from one generation to the other. And that's why there is a genetic control for gametogenesis. Now, speaking more about the role of genes in gametogenesis, we have a myriad of genes that contribute towards this symphony of gametogenesis. They help to regulate critical processes such as the meiosis we talked about, the points where the cell will differentiate, and then the maturation stages of the gametes. are all contributed to by genes. And in spermatogenesis, for example, genes such as the SYCP3, which stands for the synaptonemal complex protein 3, synaptonemal complex protein 3. So it's a protein that is involved in the formation of synaptonemal complex during the meiosis. And this complex plays a very crucial role in the chromosome pairing and recombination. We also have the DMC1, which stands for disrupted meiotic cDNA1. It's now known as the DNA meiotic recombinase 1. This is a gene that is involved in the repair of the double helix, the double strand breaks whenever there is recombination during meiosis, and it helps to ensure that it helps to promote the exchange of genetic material between homologous chromosomes. So you also have the PRM1, which stands for protamine 1. It's a gene that codes for a protein called protamine. And protamines are essential for condensation and packaging of sperm DNA, thereby contributing to how the mature and compact sperm cells will be formed. In oogenesis, on the other end, we have genes like the FOXL2. FOXL2 stands for the Fox head box L2. Okay, so it's a gene that plays a critical role in the development of the ovary. And whenever this gene is mutation, it's usually associated with disorders that affect ovarian function and female reproductive health. We also have the NOBOX gene, which stands for newborn ovary homeobox. Newborn ovary homeobox. Okay, this is another gene that is crucial for the development and maintenance of the ovarian follicles. It's involved in regulating the expression of genes that are necessary for the development of the egg. So all of these genes are quite essential for proper progression of oogenesis and development um, of of mature eggs. We also have the GDF9, which stands for growth differentiation factor 9, which is another gene that codes for protein. And of course, it's also important in the development of ovarian follicles. It's particularly involved in the regulation of oocyte maturation, and it's very crucial to the female fertility. So these genes, they act as guardians that carefully control the checkpoints to ensure that there is a seamless progression of gametogenesis across board. So speaking more about oogenesis, we're starting with oogenesis. After considering, we'll list a number of genes in the next slide. So these genes, along with these ovarian development genes, along with estrogen and progesterone, they intricately regulate the various stages of oogenesis in females. And uh, these hormones play a very crucial role in influencing the growth and maturation of ovarian follicles as we prepare them for fertilization. So here we have a list of a number of these genes, and these genes, they regulate ovarian development and they also contribute to folliculogenesis. So I have grouped them into three main groups. So you have the transcription factors. By these we mean genes that regulate the expression of other genes. That's what we mean by transcription factors. An example of them is the FOXL2 that we mentioned, the Fox head box L2. That's what is meant by FOXL2. So the main function of this, is in ovarian development, playing a very key role in promoting the development and maintenance of ovarian follicles. And so you see here listed is so it's critical for ovarian differentiation and suppression of the male pathway genes so that this really expresses a female gamete. So that's why it's very critical for ovarian differentiation. So ovarian follicles are structures within the ovaries that contain the developing eggs. Okay. Next we have the NOBOX, that's the newborn ovary homeobox, like I alluded to earlier, which is very essential for early stages of ovarian follicle development. Folks, NOBOX regulates the expression of genes that are involved in oocyte development and in folliculogenesis. That's what we call follicular development. That's folliculogenesis. So it's very essential for oocyte-specific gene expression. We also have the FIGLA. FIGLA stands for folliculogenesis specific basic helix loop helix. Basic helix loop helix. So this is it regulates expression of the zona pellucida genes, and it regulates early stages of folliculogenesis. So it's apparently involved in ovarian development, and it's expressed in developing oocytes, and it contributes to the function and formation of the ovarian follicles. Next is this category, which are known as the signaling pathway regulators. We mean genes that are involved in cellular signaling, and an example of them is the WINS4. WIN4 stands for wingless type MMTV integration site family member 4. And this gene, it's a gene that plays a crucial role in ovarian development by promoting the formation of female reproductive structures. It's also involved in the development of the Mullerian ducts, which give rise to the female reproductive tract. So it promotes ovarian development and suppresses testis formation. Next is the RSPO1, and that stands for R-spondin1. Okay, and the RSPO1 is associated with ovarian development and is involved in the regulation of the Wnt signaling pathways we just mentioned. So proper Wnt signaling is essential for normal ovarian development and the maintenance of female reproductive function. So there you have the RSPO1. Crucial for ovarian development. And then the third category is the growth factors. We mean genes that are promoting cellular growth and differentiation. And a chief example of this is the GDF9, which is growth differentiation factor 9. And this gene is quite essential for follicular growth and for the maturation of oocyte. So even though it is mentioned in the context of oogenesis, it's highly involved. I mean, the GDF9 is highly involved in ovarian development more broadly, and it's crucial for growth and maturation of ovarian follicles, and is expressed by developing oocytes. So these genes, they are largely female specific because they are crucial for ovarian development and function. So for male development, that's governed by different set of genes, which we are coming to in the next slide. While you have things like Wnt signaling, genes like Wnt and growth factors may also have minor roles in male development. So let's move on. So spermatogenesis now, the SRY gene stands for sex determining region Y. Sex determining region Y. It's a gene that is located on the Y chromosome, and it's found not only in humans. Generally, it's found in mammals, so including humans. And the SRY gene plays a very crucial role in the determination of biological sex, specifically in the development of characteristics that make one a male. So its primary gender trigger. It's the primary genetic trigger for a gender, and it helps in the development of male sex characteristics during embryonic development. And once you have it on the Y chromosome, it initiates the cascade of events that will then lead to the formation of male gonads. So there you have the SRY, and while leading to the formation of male gonads, it suppresses, the formation of female gonads. So together with testosterone, hormones like testosterone, the SRY gene is very pivotal and plays a key role in the orchestration of spermatogenesis, thus initiating and sustaining the development of a mature sperm. So here are key genes that regulate the development of the testes across spermatogenetic stages. So again, the transcription factors, which are genes that regulate the expression of other genes. You have the SRY that we just mentioned. So it's considered a master regulator of testis development, which initiates the sexual differentiation of the male sex by activating the SOX9, which we are talking about here next. So it's very essential for the differentiation of cells to becoming the Sertoli cells. And next, within this transcription factor, another transcription factor is SOX9, which is the SRY box 9. So this one helps to maintain the development of the testes by regulating the anti-Mullerian hormone. It also promotes the differentiation of the Sertoli cells and suppresses the development of the ovary. We also have, as the third gene amongst these transcription factors, is the DMRT1, which means double sex and MAP3 related transcription factor 1, which helps to maintain male gonadal identity after birth, postnatally. And it's much required for the function of the Sertoli cell and for spermatogonial maintenance. The second category of genes are the signaling pathway regulators. These are the genes that are involved in cell signaling, and this includes FGF9, which I mentioned not long ago, which promotes the proliferation of the Sertoli cells after formation, and then suppresses ovarian differentiation by antagonizing the Wnt4. It's very critical for the formation of the testicular cord. We also have the desert hedgehog, DHH, desert hedgehog, which helps to signal, so signals from Sertoli cells to Leydig cells, right, to promote their differentiation and testosterone production. So this DHH is very essential for testis cord formation as well, and for germ survival. We mentioned Wnt4 earlier, and here is Wnt5 in the male spermatogenic signaling pathway regulation. And this stands for wingless type MMTV integration site family member 5A, which regulates testis cord elongation and the development of the gonad. And then the third category, which are growth factors, referring to genes that promote cellular growth and differentiation. Chief examples are the GDNF, that's glial cell line derived neurotrophic factor, which helps to support the self-renewal, and spermatogonial stem cells. So it signals through the RET receptor on spermatogonia. We have the KIT-LG, which is the KIT ligand or stem cell factor. It's produced by the Sertoli cells, and it binds to the KIT receptor on the spermatogonia to promote survival and proliferation. And lastly, we have the BMP8B. That's a bone morphogenic protein 8B, which is required for spermatogenesis and male fertility. It strongly helps in proliferation of germ cells during development. And this BMP8B is one of the is part of the BMP family, which are a group of cytokines and growth factors that are originally discovered due to their role in inducing the formation of bones and cartilages. But of course, their role goes way beyond bone formation or bone biology. So there are meiosis specific genes as well, and meiosis specific genes help to ensure proper division and recombination of genetic materials during gametogenesis. For example, the synaptonemal complex proteins SYCP1, 2, and 3, they help to mediate chromosomal synapses, right? So they facilitate chromosomal pairing and recombination. While MLH1, which stands for methyl homolog 1, help to facilitate crossing over in order to enhance genetic diversity. So it ensures the accuracy of the crossing over of homologous chromosomes. While you have DMC1, which it stands for the new name is DNA meiotic recombinase 1. It's very essential for homologous chromosome recombination. However, this acronym was created because the original name was disrupted meiotic cDNA1. That was the original name assigned to DMC1 when it was first identified in yeast due to its role in meiotic recombination. The name has, however, it has been changed to DNA meiotic recombinase 1, because that's a standardized and widely accepted full name in modern molecular biology, because this better reflects the function of DMC1 as a recombinase enzyme that is critical for homologous chromosome pairing and recombination during the process of meiosis. So the role of hormones in this cannot be overemphasized. Hormonal regulation plays a very vital role in gametogenesis. The follicle-stimulating hormone, for example, and the luteinizing hormone, they are critical for gonadal functions in both sexes. FSH stimulates follicular growth in females, and spermatogenesis in males. So follicles, you find them in both. So follicle stimulating hormone, while luteinizing hormone triggers ovulation and it supports Leydig cell function in male. So both of these hormones are critical for gonadal functions in both males and in females. While the CYP19A1 stands for cytochrome P450 family 19, subfamily A member 1. What this does is to catalyze the conversion of androgens to estrogens, thereby promoting the development of the follicles in females. So that's why we say it converts androgens to estrogens for follicle development. We also have the INSL3, which stands for insulin-like peptide 3. And what this chiefly does is to regulate the descent of the testes. It regulates the testicular descent, while the anti-Mullerian hormone suppresses female duct development during embryogenesis. So epigenetic regulation, we have epigenetic mechanisms that help to regulate gametogenesis by modulating gene expression without altering the DNA sequence. That's why it's epigenetic. So it's not really affecting the genes like that. It's only, it's modulating the expression of those genes without altering the DNA sequence. So a number of mechanisms are involved in epigenetics, and that includes DNA methylation, histone methylation, histone modification, and non-coding RNA. So these three, they add a layer of complexity to the regulation of gene expression during gametogenesis. As for histone modification, such as acetylation and methylation, it facilitates chromatin remodeling for gene activation or repression, and that's why you have the writers, the readers, and the erasers. Okay, while in DNA methylation, which is very essential for genomic imprinting, gene silencing, and in maintaining cell lineage specificity, and that's where you have the writers and the erasers. We have non-coding RNA too, and RNA, non-coding RNA, that's ncRNA. They are RNA molecules that do not code for proteins. All they do is regulatory rules, and this category of epigenetic mechanisms includes microRNAs, which is myRNAs, and long non-coding RNAs, that's the LNC RNAs. And these and the function and LNC RNAs, for example, or let me start with the ncRNAs. The non-coding RNAs generally, they are involved in gene silencing, like I mentioned earlier, and regulation by interacting with DNA and RNA, as well as proteins. For example, this microRNA binds to messenger RNAs, and thereafter prevents the translation, while LNC-RNAs can recruit chromatin-modifying enzymes to specific gene regions, thereby influencing transcription and chromatin remodeling generally. So these epigenetic factors and modification mechanism really play a lot of role in fine-tuning the activity of genes, thereby contributing to the precise orchestration of gametogenesis at the end of the day. And during spermatogenesis, or particularly during spermiogenesis, for example, histones are replaced by protamines in order to achieve, we have protamines 1 and 2. Protamines 1 and 2. Protamines is PRM 1 and 2. So histones are replaced by these protamines protamines 1 and 2 to achieve highly compacted DNA in the spermatozoa. Here is the histone before the epigenetic modification, before they are replaced by protamines. Here is the histone. It's 100% histone at this point, and at the end of this, so these are spermatids, right? So at the end of this, when we have spermatozoa, you now have only 5% being made up of histone, while the 95% of the spermatozoa is made of protamines, as you see here. So here is an image that is helping to appreciate the key factors that are related to histone to protamine transition. So once you have the global H2A and H2B and H3 histone variants creating highly unstable nucleosomes, which is what you have here, then the nucleosomes will undergo histone hyperacetylation by EPC1 and TIP60 or some other nucleosome acetyltransferase complexes that you see scattered all around here. So acetylation at these critical lysines will then further destabilize the nucleosomes. And once they destabilize the nucleosomes, while you then have the tail acetylation, generating a platform for recruitment of BRDT, which is shown here. So BRDT stands for bromodomain testis specific. Bromodomain testis specific. It's a gene that actually encodes for a protein known as BRDT itself, which is specifically expressed in the testes. It contains bromodomains, which are regions that recognize and bind to acetylated lysines on histones and other proteins. And this function is very important in regulating gene expression and in remodeling the chromatin, particularly during spermatogenesis. So BRDT later on interacts with the SWI or SNF. SWI stands for switch, while SNF stands for sucrose non-fermentable. So the BRDT interacts with this protein, this family protein, and then starts the process of histone eviction and replacement by the TPs. So at the end of the day, you find the degradation happening, and then the evicted acetylase histone will then be recognized by PA200, and the process continues down there, until you have a spermatozoa that has just 5% histone, and 95% protamines. And non-coding RNAs could also play roles in regulating the expression of genes, that influence protamine expression, or vice versa as well, where you have protamine helping to maintain the correct chromatin structure to allow for the non-coding RNA activity. So I just added this as a epigenetic note or application. So we have a number of molecular regulators playing essential roles in gamete maturation. For example, the MOS, which is the serine or threonine or kinase. It ensures meiotic arrest in oocyte until ovulation. So it actually stands for mitogen activated serine or threonine, MOS. We also have the ZP3, that's zona pellucida glycoprotein 3, which is a critical zona pellucida protein that is involved in sperm egg binding during fertilization. So various growth factors also support the development and maturation of gametes by promoting cell division and differentiation, some of which we have noted. So you have all of this interplay going on here. So there are a number of genetic defects and disorders that could arise whenever things go wrong. So genetic and epigenetic defects can severely impair gametogenesis, thus leading to a range of reproductive and developmental disorders. And one of the key genetic deficits is found in the SYP3 mutation, in the SYP3 gene mutation, which plays a critical role, like we mentioned already, in chromosomal pairing during meiosis. So mutation in SYCP3 will disrupt this process, leading to errors in chromosomal alignment and segregation. And this can result in male infertility or gonadal dysgenesis. So where it doesn't even the gonad either do not descend or do not form well, and these are conditions where formation of viable gametes is compromised and often leading to problems with fertility. Another gene involved in meiotic integrity is the MLH1, which is essential for the proper repair of DNA during meiosis, like we discussed earlier. So when this gene is mutated, it leads to meiotic arrest, causing chromosomal abnormalities in like aneuploidy, where you have gametes that have abnormal number of chromosomes. That's aneuploidy. So euploidy is the normal number of chromosomes. So once it's aneuploidy, whether it is trisomy, monosomy, tetrasomy, pentasomy, they are all aneuploidic. So in that case, it is not normal. So euploidy in humans is going to be diploidy, so to speak, and depending on context as well. So a mutation in this will lead to arrest in meiosis, and it can further contribute to recurrent miscarriages or infertility, whether in males or in females. So it's quite important. Moreover, this same MLH1 mutation has also been associated with what is known as Lynch syndrome, which is a hereditary condition that increases the risk of various cancers, including colorectal cancers, uterine cancer, and ovarian cancer. We also have epigenetic errors, such as imprinting, where you have genes that are expressed in a parent of origin specific manner. All right. So imprinting is such epigenetic error where you have genes expressed in a parent of origin specific manner. It can lead to significant developmental disorders. For instance, the Prader-Willi syndrome and the Angelman syndromes are both caused by the loss of function of the same region of chromosome 15. But the effect differs depending on whether the mutation is inherited from the mother or from the father. That's why we say the parent of origin specific manner. Okay, that's imprinting. So it carries the source. So on the other hand, Angelman syndrome. So in the case of Prader-Willi, the paternal allele is affected, leading to hypotonia or developmental delays and obesity, while Angelman syndrome involves the maternal allele, which is defective and thus resulting into severe developmental and neurological impairment, including intellectual disability, including seizures, motor dysfunctions, and all of these disorders actually highlight the critical importance of proper epigenetic regulation for normal development. So in summary, both genetic mutations and epigenetic defects in genes are very critical for gametogenesis, and it can lead to a range of severe reproductive and developmental disorders. And this really underscores the delicate balance that is required for proper meiosis and genetic expression and regulation. So to start rounding up, we have other genetic disorders that could result from gametogenesis, like I mentioned. So this would include Turner syndrome, the Klinefelter syndrome, and others. So as for Turner syndrome, which is illustrated here, it's seen by the absence of an X chromosome in the female. So it's a female, but instead of having two X chromosomes, it has one, and they usually have low hairlines. They have short stature. They have broad chest. Such females will have curvature in their spine, which could be lordosis, or scoliosis, and in some cases could also be kyphosis. So they also tend to have widely spaced nipples. I mean, individuals with Turner syndrome. They also have elbow deformity and short fourth metacarpal. When you look at their fourth digit, their fourth metacarpal, it's usually shorter. Okay, then they have this pubertal failure, and then brown spots, known as Neville, on their limbs. They also have knee deformity. They can have structural abnormalities in the kidney. They can have congenital heart diseases. They can have webbed neck or broad, short appearing neck. they have low set ears, and they can have droopy eyelids as well. So all of these are characteristics of females that have the Turner syndrome. On the other hand, Klinefelter syndrome, which is shown here, involves the presence of an extra X chromosome. In the case of Turner syndrome, it's a female that has one X chromosome. So such an individual is going to be having a defective, an incomplete X chromosome. An individual should have 23XY, right? 23XY. So a normal gamete is meant to be 23. A normal haploid gamete is meant to be 23, which will include one X chromosome or one Y chromosome, right? So from those 23, 22 of them are autosomes, in the sense that they have genes that code for the regulation of, or the formation of, a particular function on the rest of the body, and not on the gametes. Okay, so those are autosomes. 22 of them. It's the 23rd one that is a sex chromosome. Okay, so if that is haploid, and then you have the other haploid also coming from maybe the mother, so another 23, the last of which, the 23rd of which, is also a sex chromosome. So at the end of the day, an individual has 46, a normal individual has 46 chromosomes. 44 of them will then be autosomes. That's 22 times 2. That will be 44 autosomes. And then the remaining 2 will be XX or XY, depending on whether the person will end up being a female or a male. So it means that in the case of a patient with Turner syndrome that we discussed minutes ago or seconds ago, that individual will have 44 autosomes, but one sex chromosome. So the person will end up having 45 in total, right? 45 total chromosomes, and one of them is an X chromosome. So an individual with Turner syndrome has 45 chromosomes in total, instead of 46. And in the case of Klinefelter, the person has 44 autosomes, right? And then instead of 2 sex chromosomes to make up 46, the person has 3 sex chromosomes. And then the genotype is then going to be XXY, because this person has an extra X chromosome. So an individual with such a genotype would have 44 plus 3, that's 47, and that will be XXY. All right, they are usually tall, and then so the person, because the person has a Y chromosome, that person is going to be a male. Even if you have so many X chromosomes and then there is Y, a single Y, that person is a male. Right? You may end up having a lot of feminine features. Okay, in the case of Turner syndrome, the person will be a female so long as the only chromosome available is an X chromosome, and X belongs to females. Right? So the person is going to be a female with some deficiencies. Okay, has been highlighted. So in the case of Klinefelter, the person is a male, so they're usually tall. They have tendency to lose their chest hair. They have gynecomastia, that is having a breast tissue that looks like that of a female. And then osteoporosis, they also tend to have long arms and long legs. They tend to have poor growth of their beards because of, they have a lot of feminine features, and then they have female pattern of fat distribution. Another thing that makes them look like female is the pubic hair, which is usually domiciled is usually restricted to the pubic area. So unlike in the case of a male that will have more hair, more pubic hair spread around the thighs or the perineal region, but in the case of females, it's usually domiciled to the pubic region, and that's what the patients or individuals with Klinefelter syndrome would also exhibit. They also have atrophy of the testes, testicular atrophy. That means the testis is not well grown. So a good note here is that many of these features will persist if the individuals are not treated. So many of the features are not seen when the individuals are treated, but when they are not treated, a number of these symptoms will become very apparent. And then thirdly, PCOS, that's polycystic ovarian syndrome, polycystic ovarian syndrome or polycystic ovary syndrome. So it's a common hormonal disorder in females, particularly females that are of reproductive age. And while the exact cause is not fully understood, there is usually a genetic component. And PCOS is characterized by irregular menstrual cycles. They have trouble conceiving. They could have mood changes, sudden mood changes. They tend to have a lot of acne, fatigue, insulin resistance, high levels of testosterone. They can have male pattern of baldness or thinning of their hair. They could have irregular or missed periods. They could have low sex drive. Such individuals could also have ovarian cysts, and then they may have weight changes and trouble in losing weight, and then they could also have excessive body hair growth. So individuals with PCOS tend to come with different possibility of symptoms. There are other possible genetic disorders that I didn't mention here, including the Fragile X syndrome that could affect both males and females, which is a condition where the FMRI gene on chromosome X is mutated, and in males it is Fragile X syndrome, which can lead to testicular dysfunction. So chromosome microdeletions are also possible, hemochromatosis, congenital adrenal hyperplasia, amongst others. So, as we've always tried to highlight, researchers employ a variety of advanced technologies and novel approaches, to unravel the intricacies of genetic control in gametogenesis. And these techniques have always proven to be very insightful into understanding the molecular mechanisms and the patterns of gene expression, as well as understanding better how things are epigenetically regulated during the development of these gametes. And among those approaches include the single RNA sequencing. So the single RNA sequencing actually it allows transcriptome of individual cells to be analyzed in order to have a detailed picture of gene expression at the single cell level. And this technique is particularly valuable for studying the heterogeneity within the cell populations during gametogenesis. So next is the CRISPR-Cas9, which we also discussed during our talks on induction and differentiation. So it's a very revolutionary genome editing tool that enables precise modification of specific DNA sequences. Researchers have always used this to study the functions of a particular gene that is involved in gametogenesis, thereby helping to uncover what roles those genes have in the process. We also have the ChIP-seq, that's chromatin immunoprecipitation sequencing, and this one is used to investigate protein DNA interactions in order to identify what regions of the genome are associated with what specific proteins. So this comes useful when researchers are interested in studying the binding of transcription factors, such as including histone modifications as well, or when they are interested in understanding epigenetic marks during gametogenesis. So there is also the 3D chromosome conformation capture. That's Hi-C, 3D chromosome conformation capture, which is a very powerful technique that captures the spatial organization of chromosomes within the nucleus. This one provides information about how different parts of the genome interact with each other, particularly in three-dimensional space, and that's why it's given this name 3D. So thereby offering insight into higher order chromatin structure. We also have the RNA-seq, which is widely used in profiling the entire transcriptome. So in the case of this, it provides information about how abundant and diverse the RNA molecules are. This is particularly helpful to researchers in identifying differentially expressed genes and alternative splicing events that occur during gametogenesis. So there are a number of other such research endeavors and future directions. So future breakthroughs may also lead to improved fertility treatments. Through all of this research, it will help in having more genetic therapies and having a deeper understanding of reproductive disorders. So we've understood from this talk that gametogenesis is a complex interplay of genes, hormones, and epigenetic mechanisms, and it's very crucial for reproductive success and continuation of life. And with all that we have discussed, we understand that gametogenesis holds that key to uh, addressing reproductive challenges. How much we understand and conduct research in this area helps us to identify how to make the human reproductive challenges better, and in order to advance our understanding of human development. So that's it for today. If you're interested in reading more on sources that were used in putting these lectures together, here they are listed, and I'm very happy to have had your attention throughout this. Thank you very much, and I'll look forward to having you give your feedbacks on what part of this lecture resonates with you the most in the comment section. I wish you all the very best, and until we see again, bye.